Hello, everyone, and welcome to another session on general microbiology, where we are going to be learning about the staining techniques, right? So, we've already covered the morphology of bacteria, so now we want to see how we can stain the different bacteria. So, the first thing that we're going to start with is that why do we even need staining, right? So, why do we need staining? Because we cannot see bacteria because bacteria are translucent. Right, so that is why we need the stains because bacteria are translucent, and if we want to see them under the microscope, they will not be very much visible. Seeing this picture on here, so can you see this is an unstained picture, and when I have stained this, how the contrast has increased. So all the pictures on the left are the unstained ones, and as soon as I have stained them, how much more properly they are visible. Right, so that is why it is important to stain because we want to see the structures properly so that they can be properly seen under the microscope. So because bacteria are translucent and to see them under microscope, right? So as we said, staining leads to what? Staining will lead to increase in the contrast and make the bacteria more visible. So whenever we do the staining, the first thing that we do is that we prepare a smear, right? So we take the colony and then we prepare a smear on a slide. So smearing is nothing but taking a colony and making a round colored thing. So after we have made the colony, so we can make smears either from directly the samples, or we have, if we have isolated an organism and we want to identify it, then we can take it from the colony that we have grown on the culture. So that is the way two types of smears that we can make, either directly from the specimen, the clinical sample, and the other one is that if we have isolated a colony on an artificial media, we can make the smear from that. So on a glass slide, we make that smear, and then what do? We have to fix the specimen. So we have to fix the specimen. What will the fixing do? What will the fixing do? It will basically attach, or it will kind of fix the organism on the slide, and it will also lead to what? Fixing of the internal structures. So because we want to see the internal structures, can you see here also we are seeing so many internal structures. So because we want to see the internal, we want to keep those internal structures intact. Right, we don't want them to disseminate when we are staining them, so we want to keep the internal structures intact. That is why we have to fix the specimen. So we have to fix the specimen to keep internal structures intact, right? So how do we